Gracias a Pedro y a Oscar y a todos ustedes por venir hoy en un día un poquito diferente para los seminarios. Como he dicho, eso es un poquito de un seguimiento de la charla que presentó Ingrid hace un mes y medio o algo así. Y voy a repetir un poquito de lo que ella ha dicho, pero después vamos a otro tema que está relacionado pero muy distinto. Okay, let's see. So, I'll start off by just recognizing that this work is the product of a lot of different people's efforts. Ingrid is the main collaborator for a lot of this work, and for a lot of the phylogenetic work, it's done with Cam Webb at Harvard Forest and Roger McGarry, who's with the USDA and North Carolina State University. And I'll talk about some of that work, plus a whole bunch of other people. And then move to Panama and talk about work that's going on there. And my main collaborators there are Steve Hubble, Brant Faircloth now no longer at UCLA, at Louisiana State, and Travis Glenn at the University of Georgia. These are the molecular folks with whom I'm working. And then Javier Ballesteros is the person on the ground doing most of the work. And then both in California and in Panama, there's lots and lots and lots of other people that are involved in actually collecting a lot of the data. So all the way through, this is a we project, not just me. So what I want to talk about today is thinking about where plant diseases fit into our understanding of plant community diversity and structure. And I'll start just with some, which is where I've been working for the last 25 years or so, mostly in the tropics and now more and more in Mediterranean climates. And I'll start off by some overview of the way I've traditionally been thinking about that. Then take a look at experimental and other work I've done as a plant pathologist to try to understand the idea of host range and bringing a phylogenetic perspective to that work. And then two different entries into looking at how plant diseases affect plant diversity. In the first, this is the part that will repeat a little bit and extend a little bit about what Ingrid talked about. And then I'll present that as a way of contrasting to another project looking at the disadvantage of being rare, mainly in tropical systems, but again, looking at plant diseases. So I'll start off with the work that's done between Cam Webb, phylogenetic ecologist, and Roger McGarry, who's another plant pathologist. And as a plant pathologist, when I was first trained in that, the main thing that is a given, and that you talked about the first day, is that diseases are bad for plants. Okay? That's probably pretty easy to accept. There's lots of ways in which it can be bad. As disease gets worse, the survival of plants, the likelihood of survival is likely to go down. The amount of growth that a plant does is likely to also go down. And they're also less likely to not be able to reproduce as well. So for pathogens that, like in this case, are taking away photosynthetic material from leaf surfaces or affecting plants in other ways, in almost all cases, they're going to be bad for individual plants. If we take a look at a population level, there's another fundamental idea that is embedded in plant pathology, and that's the idea that most plant pathogens, most diseases, respond to the density of their hosts. So in an area with very few hosts spread out, pathogens are less likely to spread from one individual to another. And as that density increases and they get packed together, the pathogens can spread more easily, and so you end up with greater spread, greater infection, greater disease. So it's a density responsive disease development, and that can have some implications where if we start off with a small population of plants down here, and there's a pathogen affecting it. That pathogen will not have enough hosts to reproduce on, so its population will go down. When that pathogen population is now down, the population of the plant, the dotted line, can go back up. And as that density increases, so can that of the pathogen. And we end up with these cycles of plants and their specialized pathogens, or wolves and elk, or whatever other predator-prey kind of interaction you might expect to have. We can extend that into a community where we have up here plant A, the dotted line, and its specialized pathogen A, the one that we just looked at. Then over here we've got a different plant species and its own specialized pathogen, and plant C with its specialized pathogen. And if each of those plants and pathogens in pairs are acting in a density-responsive, density-dependent manner, we can have cycling of these different species where anytime any species gets to be any plant species gets to be very common, its population will be kept in check by its pathogens. And that should, overall, prevent competitive dominance of any one of the species. And that should help keep rare species in the system. This has been proposed as a way of maintaining diversity in plant systems, first proposed by J.B. Gilbert in 1962, and then extended into an explicitly spatial version by Dan Jansen and Joseph Connell in the early 1970s. And it's since been known as a very popular idea of how plant species are maintained, especially in tropical forests, as the Jansen-Connell hypothesis. This has a number of assumptions, but among the most critical is, first, disease is bad for plants. It hurts individual plants, and as the disease increases, it affects the plant populations. But it can be good for plant diversity as long as those pathogens are host-specific. So that's a critical assumption that goes into this model. So a while back, Cam Webb and I wanted to really take a look at how robust that assumption is. So working in Panama, I walked into the last time that Ingrid and I were on a sabbatical. We spent a year in Panama and walked out into the forest, and if you wander around, leaves of everything in the forest have disease on them. So here's just a collection of 9 different very attractive disease symptoms on tropical species. And within each of those disease symptoms, there are fungi, equally beautiful and attractive, that cause those diseases. Okay? So we isolated these fungi from an array of different diseases and developed a method where we could actually test to see if those fungi that we took out of diseased leaves could cause disease back on that same host, the process known as proving Koch's postulates, Koch's proof of pathogenicity, where you take a pathogen out, put it back in a host, and cause disease. And also testing to see whether a pathogen that came from one plant could cause disease on other plants in the area around it. But we wanted to do that out in the real world, and so it took those fungi isolated, and I grew them up in the caps of little sample tubes in sterile media. So here's fungus growing, and it's reproducing here a little bit. Then out in the forest, took these little tools that have pinpricks to make very small wounds on the bottoms of the leaves, because a great many of the fungi that cause diseases in plants require little wounds caused by piercing and sucking insects or other kinds of things. Then took those cultures and attached it to the leaf of the plant using a bent clip for your hair, and then left them out in the rainforest. And after a week, if the plant was resistant to that fungus, you got essentially no response, just little wounds there. But if the fungus was able to attack and cause disease in the plant, it would cause disease. So I did this for lots and lots of pathogens on many plants. In this case, what I'll show you are the results from just 40 fungal pathogens taken from a 50-meter radius patch of forest and then tested back out onto usually 36 host species. There were a couple that, for a variety of reasons, didn't work and have smaller numbers, but usually on 36 other plant species that were all living in the same area. And what we found was, if we looked at each of those 40 fungi, that some of them were able, like this bar on the left here, were able to cause disease only on the host from which it was isolated. So it was highly specialized right here. There were 2 pathogens that only caused disease on its original host. Over here, there is 1 fungus that every single host species I put it on, it caused disease. An extreme generalist, a pathogen that can eat basically anything green in that area. Kind of scary thought that it can be that generalist. But most of those fungi are somewhere here in the middle. They're polyphagous. They can eat a number of different hosts, but they can't eat everything. Okay? And overall, quarter to a third of all the plants in this patch of rainforest were susceptible to disease caused by any particular pathogen. So a pathogen can, in fact, attack about a quarter to a third of all the plants in a particular area. Okay. The question then is, which hosts? Okay. We know they're not species specific, but is there some kind of structure to this, or is this just a random quarter of the host in an area? And that's when working with Cam Webb sort of turned to taking a phylogenetic perspective on this work. And the idea is to see if there's a phylogenetic signal in host range. And so the phylogenetic signal, the idea that closely related trees are more likely to share a trait than are distantly related species. When those traits are evolutionarily conserved, people often think about different kinds of traits of the plant that are kinds of fruits they produce, or their leaf thickness or chemistry. Here might be an example of a number of species here that share a common trait that are all in this particular clade of these 2 closely related clades, but the trait is not present in more distant related plants. I was interested in whether we can consider susceptibility to a pathogen as a composite trait and whether that susceptibility would show a phylogenetic signal. And that susceptibility is the composite of the chemistry, the physical structure, the developmental processes in the plant all put together, and how that matches with the chemistry, the physical features of the pathogens. So what I did was went back out into that forest, actually in 4 different places, did this. Found all the plants that were in that area and constructed an overall tree, phylogenetic tree of the plants in that area. Isolated fungi, this right here, this Pestalotiopsis, isolated from one of the hosts, and then inoculated that fungus onto hosts from all across the phylogenetic tree, so that we have some that are being inoculated back onto very closely related species. There's a distance matrix here, and some onto species that are very far distant in the phylogenetic tree. And we did this for a whole lot of different fungi in 4 different places, and overall this is what came out of it. I think Ingrid might have shown this slide before, but this is the way we created it. On the x-axis is the phylogenetic distance between the plant from which I isolated the fungus to the plant onto which it was inoculated. So that phylogenetic distance, 0 would be inoculated onto the same species. And this is the proportion of all the different combinations of fungi and plant at that distance that were that developed symptoms using that field inoculation technique. The symbols here, the larger the symbol, the more fungus plant species pair combinations there were at that particular 5 million year interval. And these lines, the 4 lines are not particularly important, but they show the logistic regressions of the interactions between being susceptible or resistant and the phylogenetic distance of those host pairs. And as one might expect, plants that are more closely related within the same genus, for example, have a very high probability of sharing pathogens. That decreases continuously with phylogenetic distance between those pairs. And even if you go out to far distances, plants that are in different, outside of families, so they're in the same order but different families or across orders, if you only look at those, this line shows that we still have that kind of a phylogenetic signal way out on the tail. So this is something that we would have suspected. It's more likely that a pathogen can attack 2 grasses than it could, say, a grass and a banana. But here we have what were the first quantitative measures with a nice formula, so that makes it nice and scientific, a formula that predicts what the shape of that curve should be. Okay, so wanted to expand that beyond just those kinds of pathogens that cause disease on leaves in a tropical forest. So working with Roger McGarry and other people at USDA, we used a giant database that they had created with 210 angiosperm host genera, 1,670 pathogens and other insect pests and slugs and nematodes and mites and everything else, and found that looking at the host range of what they had collected, we found pretty much the same kind of phylogenetic signal across all the different kinds of plant enemies in their host range, using just those databases that were collected from a wide variety of different sources. So we were pretty confident that this kind of a shape, this continuous curve of phylogenetic distance for host range, was a reasonably robust pattern, that it's a continuous trait, and that we might be able to use that to understand something about that host range and density dependence in more complex communities. So right now we've got, just as a quick summary, is density-dependent diseases with specialized patterns can be important. It's the common pattern seen in plant pathology, and that they could be important in maintenance of disease. Most pathogens, however, are not specialized, so that should break down some of this cycling, and that close relatives share pathogens. So that takes me on to the work that Ingrid has talked about a little bit. So the idea that if we have a whole lot of different pathogens and plants, that those species that happen to be rare at a particular time should have an advantage because of this density-dependent response, except that they're not all interacting only with specialized pathogens. So traditionally, this density-dependent effect provides an advantage to species that are numerically rare. So if a host is rare, it should have an advantage over something that is very dense and very common. But we might also see that there's an advantage of being phylogenetically rare. So that if a particular plant is not related to anything else in its community, except it's very far out, very distantly related, it's highly unlikely to be sharing pathogens or other enemies with its neighbors. And so that kind of rarity could also, in a world of polyphagous enemies, also provide an advantage to those hosts. So conceptually, this is important because if we think about these focal species here and what's happening with this species, its pathogens will reproduce on that population depending on the size of the population and reinfect it, causing potentially density-dependent responses. But it'll also share pathogens with these other hosts around it through spillover. And those species that are most closely related, here's a phylogenetic tree. D and E are the most closely related species to species C. So they are likely to contribute pathogens back onto C. Those other species around it that are really abundant, like D, will contribute more, has a thick arrow, and a species that is rare but the same phylogenetic distance will contribute fewer pathogen propagules to that. And a species like F that is very distantly related, even though it's highly abundant, will contribute very little. So this gives us a way of predicting which species are likely to be contributing the most to a particular population and looking at how isolated those species are from the rest of the community and how likely they are to be sharing pathogens with that host. Oh, okay. This is a little bit out of order. We wanted to test this, as Ingrid described, in the field. And this is very dark, but this is the UCSC campus. My laboratory is right here. And all of this, it's very dark in this picture, is forest. And down here is all grassland. And we have a diversity of habitats right on the campus, ranging from beautiful redwood forest, chaparral, grassland habitats, riparian habitats. Up here is the FERP that Pedro mentioned, my 16-hectare mapped forest plot in a oak conifer mixed forest. Be happy to talk to anybody about that work, and I'll get back to it a little bit later. But we did our work in this particular case in the grassland. One of the nice things about working on this campus is there's been so much natural history done there that we know all the plants, and we were able to construct a phylogenetic tree for all the plant species on the UCSC campus and work from that and know what the phylogenetic distances were. With our trusty field assistants, and lots and lots of them, we went into the grassland and we measured two things. One was the relative abundance of all the plant species in the field, and then we also measured the amount of disease that each of those plant species was showing. And what we found was, first, that density dependence still works. So this was a comforting finding, but it was unique in some ways, that the local relative abundance of plant species against the amount of disease that species had would increase with relative abundance. So rare species had lower amounts of disease, and those species that were highly locally abundant in a small area had lots of disease. So this graph that I've shown several times now has been shown over and over and over again within species. As the density within a species increases, you get more disease. But showing that those species that are common are suffering from more disease at the time, and those that are more rare are escaping disease. So that density dependence across species is an important contribution that hasn't been shown very often. At the same time, we have evidence for phylogenetic rare advantage. Here, this is the phylogenetic distance from, again, those same species. Each dot is a different plant species in the meadow. And those species over here that have a lot of close relatives in the community also have a lot of disease. Those species over here that are phylogenetically distant from its neighbors in the community, does not have very many close relatives, have less disease. So here we have there's two components of rare species advantage. That there's a density dependence across species, so there's a numerical rare species advantage. And we know that plants share pathogens with relatives, and so those species, if we can look at how close they are to the community around it, also have an advantage if they can escape sharing pathogens because they don't have close relatives. So, so this was all in agreement with what we would have expected and predicted based on Gilbert and Jensen Connell and those kinds of density-dependent based models for the potential for pathogens to maintain species diversity, with the exception of now we have to think about more complex species networks. But maybe it's not always like that. And so this is where we branch off into a completely new direction. And this is thinking instead about if there are disadvantages that rare species have. Not necessarily a disadvantage of being rare, but that rare species might have another disadvantage. And this is part of ongoing work with Steve Hubble and Travis Glenn and Brand Faircloth and Javier Ballesteros. And this is ongoing work. So we're still in the process of analyzing and thinking about what this means, but I wanted to share with you what we've done so far and very much look forward to having more conversations. So now we go back to Panama, to Barro Colorado Island. And Barro Colorado Island, BCI here, is a wonderful place to work. The Smithsonian Tropical Research Institute has been running the island, sometimes together with other people for about getting about 90 years now, I guess. And it's an island in the middle of freshwater Lake Gatun, right in the middle of the Panama Isthmus. This is the Panama Canal that goes by right here. The dark green area is the old Panama Canal Zone, which has been maintained in forest when it was under control of the U.S. military. It is now, of course, completely under control of Panama, but it is largely now national parks and protected that way. The island is about 1,500 hectares. It's got about 2.7 meters of rain a year with a seasonal, semi-deciduous tropical forest. It has a dry season of about 3 months. It's a great place to work for many reasons. One, it has a laboratory where you can do microbiology. About half of the island is old forest that has not been disturbed by humans in at least 500 years. It has another part of the island where the forest has been disturbed 100 to 150 years ago. And Steve Hubble, Robin Foster, and put together this 50-hectare plot right here on the top of the island where we can work for studying tree dynamics. They put this together in 1981. It's a kilometer by a half a kilometer, on which there are about 313 tree species, about 250,000 individuals, everything bigger than 1 centimeter in diameter. And every 5 years since then, all those 250,000 individuals have been remeasured, recensused, new individuals mapped in, and follow through over time. So this is a tremendous resource for looking at dynamics and spatial patterns. And it was the first such plot of this scale and this design. But now there's a large number. There are 46 plots scattered all over the world as part of the Global Forest Observatory Network coordinated by the Smithsonian. Originally, it was called the Center for Tropical Forest Science, but now we're expanding out into a large number of temperate zone areas. You can see the plots are all over the place, and the most important one is right here in Santa Cruz, which is the FERP that I pointed to before. It's a slightly smaller plot. Instead of 50 hectares, it's 16 hectares, but it's part of this whole network. What I want to point out is that we have this little plot here in Santa Cruz, California. Coastal California is a Mediterranean climate, but the real Mediterranean is completely empty from permanent plots. So if you're interested in thinking about permanent plots in Mediterranean forests, which would be a real benefit to expanding this network. Let's talk. I'm on the first floor in the group office, and please come see me. So using the data from all of this network, about 4.5 million trees, 8,500 species, we know that tropical forests in particular are very species rich. And lots of people have been asking, why are there so many species of trees in tropical forests, and how do we maintain that diversity? Well, beyond just how many kinds of species, how abundant they are is also quite interesting. And for instance, on BCI, if we look at the number of stems, and this is larger than 20 centimeters in diameter, so slightly bigger trees now because we're sort of shifting into adult trees. Number of stems that are on the plot in bins of 20, bins of 20, I guess, yeah, on the plot. And the number of species, you see that most of the species right here are very rare. Most species, 115 of the species or so, have fewer than 20 individuals in the entire 50 hectares. There are a few more that are slightly more common in here. And then there are just 3 species out here that are very, very abundant, that dominate numerically the number of species that are in the forest, number of individuals that are in the forest, with just 3 species. If we expand that to look at all the CTFS plots, this is a general pattern that we see. 4% of those most abundant species, those species that are really dominant, make up about half of all the individual trees. And the rarest 50% of the species make up only 2% of all the individuals. So this is a really strong distinction between how much of the biomass is in a particular species. A few species have most of the biomass, and then the rest of it is divided up into many species that are rare, and generally tend to be rare everywhere. So the question now goes beyond why are there so many tree species, but why are there so many rare species in the forest? Why don't those rare species take advantage of being rare, of not having conspecific competitors or getting diseases or herbivores that are keeping their populations in control? Why are they not increasing? So Steve Hubble pulled together a bunch of the data from the last 30 years of plot data on BCI, and looked at some of that density-dependent effect with a particular measure. And this is the measure of how much does the growth rate of individual stems change if you add another individual of the same species. And this is just looking, not experimentally, but just looking at them across the plot. How fast is the species growing, whether it has a bunch of one neighbor very close or more neighbors? What's the effect of adding neighbors to the growth rate of individual trees? And so if we look over here at this dot right here in the bottom left-hand corner, this would be a small tree, a 1 centimeter diameter tree, juvenile. And we're looking at adding another individual of the same species within 0 to 5 meters away. So close by, another individual being added, and the individual we're interested in to see how fast it grows is small. And we can see that it has a very strong negative effect on growth of that individual. So reduction of 70% on the growth. If we look at a slightly bigger tree, 2 centimeters, now the impact on that species is not quite as great, on that individual is not quite as big. It's about half reduction. And that as those focal trees get bigger and bigger, the impact of having a conspecific neighbor gets less and less, so that by the time a tree gets to be about 20 centimeters in diameter, there's no impact of adding another neighbor of the same species nearby it. So this impact is very strong, but it's strong for small trees. if you then look at large, and this is only within 0 to 5 meters, if you look at different distances, you see that even if you take a small tree, a 1 centimeter tree, and you add neighbors to it, but now from 5 to 10 meters away is the red dots, or 10 to 15 with the blue dots, that effect of neighbors decreases very rapidly with distance, so that by the time you get up to about 20 meters away, there's no impact of having additional neighbors. So those impacts, the Jansen-Connell type impacts, or competitive interactions with negative density-dependent responses, are focused on small individuals and at a small spatial scale. Okay. So Steve played around with some other data in a different way, and instead of looking at growth of individual trees, looked at population growth rates, population growth rates across the plot in 2 different time periods. First from 1985 to 1990, second from 2000 to 2005. These are for 3 different species, Trichilia, Eugenia, Concordia, Lasiocalyx. We had these maps for all the species on the plot. And these are heat maps where the red areas show places where the population growth rate is positive during that 5-year period. And blue here is where the population growth rate, the number of individuals in that area, is negative during that time. And green is no change in the population during those 5-year periods. And so it's, what's interesting to look at in these heat maps is here on this particular map, or let's look in the middle here, you can see here's a hot spot where the population is growing rapidly during those periods, and here's another spot, and here's another spot. If you then move to 2000, 2005, there are also hot spots of the population growing and areas where the population is decreasing, and they're not in the same place. They're changing over time. And this happens for all the different species, where the places that are really good with lots of population growth right now, a couple of decades later, switch. Those are now no longer growing. They might be decreasing or staying the same. And now you have a different place where those populations are coming up. And we liken this to avena boiling in a pot, where you've got bubbles popping up in different places over time and then disappearing. And that's what's going on with these populations spatially across those 50 hectares of all the different species. So something is regulating or something is driving very locally differentiated rates of growth, positive growth and negative population growth, that's varying over time at the scale of decades. So that got us thinking about all the work that me and many other pathologists have been doing for many years on density dependence and rare species advantage. And Steve Hubble has put forward this idea that he's calling the enemy susceptibility hypothesis. And in the very, very short version of that, the idea is, what if all this stuff about density dependence, which is happening at a very small spatial scale, but that's really not what's keeping so many of those species rare. But what if instead those species are rare because of particular traits that they have that make them more susceptible to pathogens? That those trees are, and this is primarily thinking about trees that are 20 centimeters in diameter and bigger, are more susceptible to pathogens so that they can never become common. Now, there's some evidence for this kind of a thing from seedlings and seedling diseases. There's stronger soil feedback for pathogens and lower survival of rare species on the plot, looking at things in seedlings that Liza Comita had done and Scott Mangan and his co-workers have done. So at the seedling stage, there's some evidence, but it tends to be either not mechanistically tied in with diseases, or with very few species. And almost all the work that any of us have done interested in diseases in forest systems have been looking at foliar diseases or seedling diseases or things that affect small seedlings. These are mainly ascomycete fungi, some oomycete pathogens causing damping off. But big trees find most of those kinds of pathogens to be an annoyance, but not real threats. And instead, it's these kinds of fungi that are important for affecting the mortality, growth, reproduction, things like that, of larger trees. These are wood decay poor fungi. They are all basidiomycetes from actually 3 families of basidiomycetes, so a small group of these wood decay fungi that cause this hollowing out of trees. They tend to have these kind of conch-type shapes when they can do that. And they're very special because they are the only organisms that can degrade lignin directly. And tree wood is full of cellulose and hemicellulose, which is really easy for anything to eat. But it's also completely packed with lignin, which only these groups of fungi are able to eat. Bacteria, ascomycete fungi, termites, ants can't eat it directly. These fungi have to come in first, break that down, and then all kinds of other things can come in. So if you have a hollowed out tree and you've got a bunch of termites in there or other kinds of fungi growing in there, they're following these wood decay poor fungi. The other thing to keep in mind is that they're actually eating dead wood for the most part. They're eating the xylem on the inside of the tree, which is dead wood, and only some of them are able to actually attack the living wood around the outside. So it's structurally important for the trees, but it's not necessarily going to kill the tree directly. Most of these fungi come in through the base, through wounds or other things through the roots, or through root grafting between different individuals. They can cause root rot, butt rot at the base, and then go up through the column and cause heart rot, decay out, hollow the inside of them off. And what this does is it predisposes them to snapping off or falling over in rainstorms and windstorms and things like that. It's the main cause of mortality of larger trees, that it facilitates falling down in storms. So, if you walk through the forest, here are 2 trees, and ignore for the moment these implements of torture. I'll get back to that in a second. But you can see on this tree on the left that there's a hole in it right here in the middle. So you might think that tree is probably hollow. Okay, and you can take a flashlight, look in, and the tree is hollow. So there's good sign on the outside. This tree over here, you can walk all around it, you can tap on it, you can look at it. There's no sign that it's anything but a healthy tree. It's a perfectly healthy tree. But if we attach these instruments to the trees, we can see that the insides of the trees are actually both hollow. So this is a sonic tomograph of each of those trees. Brown and black is healthy tissue, and if it were a completely healthy tree, it would be brown and black all the way across. Green and magenta here indicates decay, a decay column inside the tree. So both of these trees, one with external symptoms, another without, are both decayed on the inside. These work by putting, we use very fine tacks now, but it was originally designed to use with these nails, a dozen nails just barely through the bark and into the wood around the tree, to which each is attached a sonic sensor that is connected to a computer. And then we have a $2,000 hammer with an accelerometer in it that's also connected to the computer. And you go around and take, go to one of those nails and tap it with the hammer. The hammer makes a sound that transmits through the wood, and the computer records the exact second that the hammer hit the nail and the exact millisecond that each of the other sensors heard that tap. So it knows how long it took to get from one place to another. And we actually construct very detailed geometries of these trees, so you know exactly what the distance is between those two things. And it then calculates how long it should take sound to go through the wood if it was equal in all directions. And so the deviation from that picks up places where the structure of the wood slows down the movement of sound. And so anytime it's decayed or there's a crack or something like that, sound goes through more slowly than it does through the solid wood. And we can construct these kinds of tomographs. So this is a tree here. These are 3 scans from the same tree, Guarea guidonia. And they are taken at the base of the tree. Remember, these fungi come in from the base. At a meter up, where you can still see a decay column happening. And then by the time you get up to 2 meters, that tree is solid. And this is the pattern that we see in most of the trees. Decay at the bottom, and it goes down. Sometimes it'll go up many meters, but in general, it's declining as it goes up, and it's at the base. So we have scanned so far 1,750 different trees from 173 different species, 49 families, trees from 20 centimeters up to a meter 0.3 in diameter. We can't do the really big trees for logistics reasons. And they've been sampled from all over the 50-hectare plot. And they were sampled from this odd kind of pattern in such a way that we sampled every individual of all the rare species and then subsampled from all the more common species along the way. So it's an odd kind of sampling, but it gives us a really good measure of what's happening across all the different diversity. And we can ask 3 things. One is, as the diameter of the trees gets bigger, as these trees get older, do we see more heart rot? This is something we would expect from the literature, but it's sort of a test to make sure that our method is actually picking up what's going on. But more importantly, we can say, do we see a rare species advantage, which is what you would expect if there were these density-dependent, species-specific things going on, so that abundant species have more decay, or the enemy susceptibility hypothesis that says those species that are rare should have more heart rot, and those that are common should have little. So for the first hypothesis, these are pictures that are scaled according to their real size, and we can see that small individuals of Gustavia superba have essentially no disease, and that as these trees get bigger, they become hollow. And this is a pattern that we've seen for across within species and across species. This graph is across species, where in 5-centimeter diameter classes, combining across all the species at those diameters, we see that small trees at 20 centimeters have about 12% of the stems are decayed. And this goes increasing up to trees that are 60 centimeters or more, have about 60% of them are internally hollow. And one thing to note is right here, trees reach the canopy height at which they can reproduce at about 30 centimeters dBH, and about a quarter of all the trees are already hollow by the time they have reached reproductive age across all the species that we've tested. One thing to note is I actually, decades ago, spent a whole lot of time studying these same fungi, but just using their reproductive structures on the trees. And it turns out that only about 12% of all of the trunks that are decayed show any external signs of fungi at all. So any of the studies that we would do by actually going and collecting fungi that you can see probably don't give us much, because it takes them decades before they can reproduce. So then the question about rarity. The x-axis here is the abundance of each of the different species on the 50-hectare plot. The little blue hash marks are those that are, in this case, sound, healthy trees. The ones up at the top are the ones that are decayed. And these are only for trees that are 20 to 40 centimeters in diameter, so the smaller size classes. And we have a significant decrease where for the abundant species, it's about half as likely for the species to be decayed as for the very rare species that are only found fewer than 10 times across the whole plot. If you get rid of these 3 species, you get approximately the same curve. So it's not just driven by these 3 really common species. That the rare species have a much higher probability of decay than do the rare species. But by the time those trees get to be big, that effect disappears. So that this is actually not significantly different from flat for trees bigger than 40 centimeters, where there's no difference in the decay rate depending on the abundance of the trees. The common ones are just as likely as the rare ones to have decay at that time. And that's a function of losing the rare species quickly from the system. So if we look at rare species here, and we start off with 82 species that are rare, fewer than 10 stems across the whole plot, in the small size classes. By the time you get to be 60 centimeters tall, there's only 13 species left. Only 16% of the species that are present in these smaller size classes get to be large. And in fact, it's only about a quarter that get to be reproductive size. If we look at the common species, there are 46 that have more than 100 stems, and a third of those are still present at 60 centimeters. So basically what's happening is these rare species are dying out of the system before they ever get to be big. Whereas the common species are those that are able to maintain in the system. So overall, what we've got so far, and again, this is ongoing work, is that susceptible species die sooner. Big trees are more likely to be common species because of that. Bigger, older trees are likely to get more heart rot. And the rare species are appear to be much more susceptible to this kind of important kind of pathogen than are the common species. They're about twice as likely, and that is consistent with what we see with the enemy susceptibility hypothesis. So now we've got 2 contrasting patterns. The work that Ingrid and I have done, and many others as well, on these rare species advantage. And there are lots and lots of examples now of this being important for juveniles and herbaceous plants. This is really the first study like this for long-lived trees, for bigger trees. And here it appears that there might be a rare species disadvantage. And so what we're trying to do right now, and I'm anxious to talk to people here over the next several months about this, is figure out how to reconcile these things and figure out when one might be more important than the other, and how those things, not necessarily mutually exclusive, but fit together in the maintenance of diversity in tropical forests and other systems. So again, it has been a wonderful several months here, and I'm really looking forward to the next 6 months that we have here. Everybody's been so welcoming with Pedro's lab and in Montse's lab. And had great interactions, and look forward to talking more with all of you anytime. So thank you, and I'm happy to take your questions.